acid, we can simply talk about these guys being information storage. They are the ones that are able to determine the kind of proteins that will be produced. They are the ones that are able to determine the impact of a certain virus, an impact of a certain bacterium when it is introduced in the human being. So basically, when you talk of the nucleic acid, when you talk of their function is just to store and transmit genetic information. You need to look at your family and you're going to understand that there are some genetic characteristics that you had to inherit from both your parents. This can easily be explained when you talk of human anatomy, where we had to discuss issues of the stages of the meiosis one, and one of the important stage we had to talk about is the prophase one. And under prophase one, we had to talk about issues of exchange of genetic material, which normally takes place at the packaging stage of prophase one. So all that is something that can help us to understand that when you talk of nucleic acid, we are just talking about something. We're just talking about a compound which stores what? Genetic information. And because this compound stores the genetic information, this simply means that it's helping to transmit the genetic information from the parent to the what? To the daughter cells, who are us as children. So the major function of nucleic acid, it is what? To store and transmit genetic information. But this is not the only function of this nucleic acid. So when you talk of this nucleic acid, they exist in different types. Basically, we have two types of nucleic acid. We have RNA and we also have what? DNA. So RNA is classified into three groups. You have rRNA, mRNA, tRNA. You need to understand that the rRNA and mRNA, they are being created or they are being synthesized in the nucleolus of the what? Of the cell. You know a cell the way it is, you need to understand that there is a nucleus there. When we apply an atom, we are going to understand that the nucleus is the only organelle which is not in the cytoplasm. Why? Because this guy is being bound by two membrane structure, and the outside, it is the what? The nuclear envelope. As a result, we say this nucleus is something that is not in the cytoplasm. So inside the nucleus, you have the nucleolus. So RNA is being synthesized in the what? In the nucleolus. And in the nucleolus there, you have the genetic information which is being stored by the what? By the DNA. Okay, so when you understand this, you need to understand that when DNA is being replicated, it will produce two strands. There is a leading strand and the lagging strand. So one of the two strands will be used for DNA transcription. And during DNA transcription, that's when you expect RNA to be produced. But in this case, the only information that we need to know is that there are basically two types of what? Two types of nucleic acid. You have one, DNA and RNA. These two are playing a role in the storage of what? Genetic information. And they are playing a role in the very important theory which we normally call the central dogma. So when you talk of the central dogma, it's just trying to explain how DNA is used to form a protein. Now, this is what we just need to know at the moment. But we also need to add up another component which is said to be a nucleic acid. And this is what? ATP, okay? Which is known as the adenosine triphosphate. So when you talk of ATP acts as a source of energy. And this ATP, it normally has two components. It has the adenine, which is being bonded with what? 3-phosphate. Okay, so we'll look at that later on in detail. What is the structure of this nucleic acid? That's one question we need to expect. So when you talk of the structure, nucleic acid have three parts. The first part you can talk about is what? Is the first part we can talk about is the nitrogenous base, then you have the phosphate group, and also you have the ribose sugar. Okay, so that's some of the things that you can talk about. So I don't know if you guys are able to get me, and I hope you guys are able to understand. Are you guys able to get me? All right, thank you, Christians. So when you talk of the structure of nucleic acid, like I said, nucleic acid are made up of three components. You have one, the nitrogenous base. Two, you have the what? You have the phosphate group. And three, you have the ribose sugar. Okay, and this ribose sugar is in the furanose form. Why? Because it has 5 carbons in its chain, in its ring. So we'll talk about that more in detail. So the monomers of nucleic acids are just known as what? Nucleotide. The monomers of nucleic acid are just known as the nucleotide. These nucleotides, they come together, they bond together through the help of what? Phosphodiester bond to form what? To form DNA or RNA. So all these things will be explained later on. So when you talk of nucleotide, like I said, these are what? These are basically the monomers of nucleic acid. When you talk of nucleotide, these are just the monomers of nucleic acid. Now, these nucleotides, they have three parts. Nucleotide have three parts. The first part is what? Is the nitrogenous base. The first part is the nitrogenous base, the one which has been circled there in red. I hope you are able to see it. So the nitrogenous base, this can either be purine or pyrimidine. A nitrogenous base can either be a purine or pyrimidine. So that's a nitrogenous base. The other component of the nucleotide, it is the pentose sugar. The other component of the what? Of the nucleotide is the what? A pentose sugar. And this sugar, it has what? It is a 5-ring structure. Okay, so the pentose sugar, it is the very important sugar which is found in what? In nucleotide. And it is this pentose sugar which determines whether we have RNA or DNA. RNA, you need to understand that we have what? A ribose sugar. And then when you talk of DNA, then we are talking about a deoxyribose sugar. So that's just a pentose sugar which I've circled there. The other part which is very important, when you talk of the nucleotide, it is the what? The phosphate sugar. The other type which is very important, it is the what? The phosphate sugar. So when you talk of the phosphate sugar, this is just a compound which has one phosphate group and how many? 4 oxygen molecule. One phosphate group and 4 oxygen molecule. And then what you need to understand that only one oxygen shares a double bond with a phosphate group. Only one oxygen shares a double bond with a phosphate group. The 3 oxygen, they don't share any double bond. So that circle is just simply showing the phosphate group. The circle which I've illustrated there, it's just showing the phosphate group. I hope people are able to follow, and I hope my network is allowing. Do you have any question here? Any question? Do you have any question? Hey, no question. Or I've been kicked out.
No question, sir. No question.
Is my screen visible? Or it has paused? It has paused? But my voice is okay.
Yes.
Yeah, you're loud and clear, and your screen is just fine. So I'm thinking maybe it's just their network.
Okay, it's now moving. Okay. Alright guys, so remember 3 parts of nucleotide. 1, you have the nitrogenous base. 2, you have the pentyl sugar. 3, you have the what? You have the phosphate group. Those are the 3 things that makes up a nucleotide. I'm trying by all means to summarize everything, but I'll make sure that we are a little bit in detail. Okay, so let's quickly look at RNA and DNA. Remember, the pentyl sugar, it is the one which can either give us a ribose or a deoxyribose, depending on the presence or the absence of the OH group on carbon number 2. So guys, it is, when you talk of biochemistry, it is knowing every detail. It is making sure that you know every detail. So when you talk of RNA, this RNA is normally has a single nucleotide chain. RNA has a single nucleotide chain. What about DNA? So you need to understand that when you talk of DNA, DNA is something that we can say it is a little bit double helix. It is just double helix. In short, it has 2 chains of nucleotides, and these 2 chains, they normally twist. These 2 chains, they normally twist, leading to the formation of what we are calling the double helix structure of DNA. Okay, so let me quickly